Dear students, in this class we are going to learn about nucleic acids. Nucleic acid, just like other biomolecules like carbohydrates, proteins, lipids, etc., are essential biomolecules that act as the basic molecular basis of life, and it controls the structure and function of organism through genetic information and the expression of genetic information. So as we said, chemically, nucleic acids are essential large biomolecule that act as primary information carrying molecules in cell. And as we know, they are of two types. You might have heard of them: DNA, which is deoxyribonucleic acid, and RNA, which is ribonucleic acid. Now, nucleic acid, on undergoing alkaline hydrolysis in the presence of aqueous ammonia or barium hydroxide, or enzyme hydrolysis in the presence of enzyme nuclease, give what is known as nucleotides. So, nucleic acids are essentially long chain polymers made up of nucleotides. So, in other words, they can be called as polynucleotides. Now, let us see what nucleotides are. Now, when we carry out the complete hydrolysis of nucleotide, this is broken down into a heterogeneous base, which could be a purine or pyrimidine base, a pentose sugar, and a phosphate ion. Just like this is a heterogeneous base that is linked to a pentose sugar, a sugar containing 5 carbon atom or 5 ring members, and then that is linked to a phosphate ion. So, if nucleotide is a complex molecule containing a heterogeneous base linked to a pentose sugar linked to a phosphate ion. Now, let us go more deeper into the structure of the basic structure of polynucleotide. Actually, polynucleotides are long polyester chain which is made up of, as we said, phosphoric acid as the acid moiety, sugar containing the alcohol moiety, and the heterogeneous nitrogenous heterocyclic base, which are the purines or pyrimidines attached to the ester chain at regular interval. So if we look, we have sugar. These sugars contain a lot of OH unit, that is the alcohol moiety. The phosphate ion contains the phosphoric acid moiety, and there is a linkage between sugar phosphate. Again sugar phosphate. And on the sugar there are nitrogenous bases. So polynucleotides are actually polyester chain which contain phosphoric acid units, sugars with nitrogenous bases attached at regular intervals. I hope the concept of polynucleotides are clear to you. Now, classification of nucleic acid. On the basis of the sugars present in the nucleic acid are divided into two: ribonucleic acid, RNA, deoxyribonucleic acid, DNA. And as the name indicates, RNA contains ribose sugar, and the sugar in deoxyribonucleic acid is 2-deoxyribose. This is the structure of ribose and this is the structure of deoxyribose, 2-deoxyribose. So let us concentrate on these are both Furano structure. In the case of ribose, draw a 5-membered pentagon with the oxygen at the top and the carbon right to the oxygen atom is numbered as 1. There is an OH on the upside and H on the downside. On the second carbon, the H is on the upside, OH is on the downside. On the C3 carbon also, we have OH on the downside, H on the upside. On C4 carbon, we have the H downside and CH2OH on the upside, and this CH2 is the 5th carbon. So, in the ribose sugar, please learn the position of OH, where OH, H and other atoms come. So, in the case of ribose, it is C, OH, H, C2, HOH, C3, HOH, C4, CH2OH, H. Coming to deoxyribose or 2-deoxyribose to be more correct, again draw a pentagon containing oxygen at its apex. The C1 carbon contains OH and H. C2 carbon contains both H. One OH here is replaced by H. C3 is HOH, similar to ribose. CH2OH, H at C4 carbon. So, learn to draw the structure of ribose and deoxyribose sugar. Now, these sugars, now we have a basic idea about the structure of the sugars, and these sugars form phosphate ester through the C3 and C5 carbon, C5 hydroxy groups. C3 and C5 carbon forms the phosphate ester linkages, and the bases, nitrogenous bases, are attached to the C1 through a beta glycosidic linkage. This is the beta glycosidic, means CN linkage. So, in the case of here, if we are looking at the RNA base, RNA, this is the ribose. Ribose is characterized by the presence of an OH in the downside of C2 carbon, and the C3 carbon contains a phosphate ester. The C5 carbon contains another phosphate ester, and the chain continues like that. The C1 carbon contains this nitrogenous base, which is adenine. Now, coming to the heterogeneous heterocyclic bases present in nucleic acid, it contains 2 types of bases. One is pyrimidine. We know pyrimidine is a heterocyclic aromatic compound which contains 6 members in its ring. It contains 2 nitrogen atoms at position 1 and 3 in the 6-membered ring. Purine base is a fused heterocyclic compound containing a pyrimidine ring fused to an imidazole ring. You have studied heterocyclic compounds. So this is a pyrimidine ring and that is fused to an imidazole ring. So study the structure of both pyrimidine and purine. Now this is the base of the nitrogenous bases. Coming to the original nitrogenous bases, purine bases are 2 types, that is guanine and adenine. In adenine we have a amino group in the C4 carbon, adenine in the C4 position, amine at the C4 position, and in the guanine you have at the C2 carbon we have an amine, and at the C4 we have a carbonyl group. In the case of pyrimidine base, you have three bases: uracil, cytosine, and thymine. Uracil is, you write down the structure of pyrimidine and then draw carbonyl group at 2 and 4 position. Omit one double bond, you will get the uracil. When carbonyl group at the 4th position is replaced by amino group, you will get cytosine. Then thymine is similar to uracil, but it contains a CH3 group at the 5th carbon. Okay, so there are two purine bases, adenine and guanine. There are three pyrimidine bases, uracil, cytosine and thymine. So these are the heterocyclic nitrogenous bases present in the nucleic acid. Now, coming to the RNA and DNA. RNA, that is ribonucleic acid, contain 4 nitrogenous bases, which are uracil, adenine, guanine and cytosine. Uracil, adenine, guanine and cytosine. Whereas DNA consists of thymine, adenine, guanine and cytosine. So here, if we look carefully, in RNA, uracil is present. So uracil is present only in RNA, whereas in DNA, only thymine is present. No uracil. So that is very important. RNA contains only uracil and DNA contains only thymine. And the pyrimidine bases present in DNA are cytosine and thymine. Purine bases are guanine and adenine. Similarly, in RNA, the purine bases are adenine and guanine, but the pyrimidine bases are cytosine and uracil. Now, when we study nucleic acid, we often come across two terms, nucleoside and nucleotide, which might come as a 2-mark or a 4-mark questions. So let us have a deeper understanding of what a nucleotide and what a nucleoside is. Actually, the nucleoside refers to the base sugar unit. It is the combination of nitrogenous base and sugar. And whereas nucleotide is a combination of base, sugar and phosphoric acid unit. So I hope it is clear. Nucleoside is a combination of base and sugar. Nucleotide is a combination of base, sugar and phosphoric acid unit. Now let us go through each of the nitrogenous bases. So if we consider a sugar ribose, so this is ribose. Ribose combines with the adenine. This is adenine, if you remember the structure of adenine. Ribose combines with the adenine through the C1 by a beta glycosidic linkage to form a nucleoside. And this nucleoside is known as adenosine. And this adenosine combines with the phosphoric acid at the C5 carbon, CH2OH. That OH combines with phosphoric acid to form the phosphate. And this combination of adenine, ribose and phosphate unit gives us a nucleotide. It is known as adenylic acid, or it might be more familiar as adenosine monophosphate. This is adenosine monophosphate. So this for adenine and ribose, the nucleoside is adenosine and the nucleotide is adenosine monophosphate or adenylate acid. Now, if we consider guanine, so guanine with ribose is guanosine, which is a nucleoside, and then this combines with one phosphate unit to form a nucleotide known as guanylic acid, which is a guanosine monophosphate. And if we add one more phosphate, again it is a diphosphate. If we add 3 units, it is a triphosphate. So, nucleoside triphosphate is actually a nucleotide. So, this is just to bring out the difference between what a nucleoside and nucleotide is. Now, if we consider uracil, uracil combines with the ribose to form uridine nucleoside, and this combines with the phosphate group at the C5 carbon to form uridylic acid, which is a nucleotide, uridylic acid. Now coming to cytosine. Cytosine with ribose sugar is cytidine, which is a nucleotide. That cytidine combines with the phosphoric acid unit gives a nucleotide, which is cytidylic acid or cytidine monophosphate, CMP. So in this class, we have studied the basics of the very elementary idea. We have obtained a very elementary idea about nucleic acid, what are the components of nucleic acid, nucleoside, nucleotide, etc. In the next class, we will go into the details of the structure of nucleic acid. So, until then, bye.